Hello, good evening.
Good evening, thank you, sir.
Okay, so welcome to today's tutorial. We are going to continue from where we left last time. But before we continue, we are going to have a brief overview and recap of what we looked at last time, so that our friends may have an idea and also catch up on what we are looking at currently, or what we'll be looking at today in proteins. So we began already looking at this topic on proteins, the macromolecules, proteins. So our objectives for the lesson is for us to be able to describe what proteins are, be able to define or understand the functions of the proteins, and also to understand the relationship that exists between the amino acid sequence and also their three-dimensional structure. And so the other objective is for us to be able to describe the possible levels of protein structure. So last time we did say that proteins are actually the most diverse group of biological macromolecules, both chemically and also functionally. We went on to talk about the functions of proteins. After the introduction, after an overview of what proteins are, we talked about the functions that proteins do in organisms. Those that were present, what are some of the functions of proteins that we talked about? Guys, any functions of proteins? Or anyone? Guys, what are functions of proteins?
Proteins are an enzyme catalysis, and they defense, they transport, they support, and they have regulation again.
Okay, thank you very much. So that's correct. And why were you fearing to talk? Yeah, so we said that they act as catalysts, as he has said. They also function as transport molecules. We give an example of hemoglobin in the human body. They also act for regulation, storage, support, and also for defense. And then we talked about the amino acid structure. We talked about the amino acid structure. So I said that the amino acids, which are the constituents or the monomers for proteins, have an amino group and a carboxyl group. So they have an amino group giving the name amino. And then we have the carboxyl group or carboxylic acid group. So which renders this amino acid the name acid. And then say that it has a hydrogen atom. And then we have, it also has a side chain. So this side chain can range from the simplest amino acid, which only contains an H as its side chain. Which amino acid is this one? Which has a side chain of H, just H? Guys, what's the simplest amino acid? Where the R group is, the R is simply a hydrogen atom. It's glycine. So it's amino acid, glycine. And then we said that amino acids occur in solution in their ionized form. This is showing the non-ionized form, which is the same as this above. So you have the amino group and the carboxyl group. And then this is the ionized form of the amino acids, the ionized form of the amino acids. So amino acids occur in solution at neutral pH like that. They have dipolar ions. They are dipo at neutral solution. So they are dipolar ions. They contain dipolar ions. Then we talked about the side chains or the ARA, the side chain. So these are some of the examples of the amino acids, or rather these are examples or representations of the 20 amino acids and their categories. So the simplest amino acid, which has hydrogen atom as the ARA group or the side chain, is glycine there. That's a glycine. So you have non-polar side chains, we have the polar side chains, and we have the electrically neutral side chains. Those who are there, who can recall what we said, how you were able to determine if an amino acid is acidic or it's basic or it's uncharged polar, or whether it is a non-polar amino acid? What did we say? Just looking at the same diagram there that is showing. Anyone who remembers? So we said you look at the side chain. Okay, you can say. Someone
Yes,
wanted.
you're talking about you say the R group, but you look at the R group and then you see if it has gained or lost a proton. So for this case, for acidic acids, amino acids, they always have a negative in the R group, and for basic, they have positive.
Okay. Yeah, so you look at the side chain. Thank you for that. That's correct. You look at the side chain. Does it have a negative charge? As in this case. So this is a charge chain. Remember, each and every amino acid has a same basic structure, and they only differ in the side chain. So you look at the side chain. Does it have a negative charge? If it has a negative charge, it means that this amino acid has lost a proton. It has lost a hydrogen, a proton. So this means that this is acidic. Conversely, or on the other hand, you look at the side chain. If it is positive, if it has a positive charge, it means that it has taken a proton. And so this is a basic amino acid. It's basic amino acid. However, if the side chain is uncharged, you look at it's the same side chain and see if there is an oxygen atom. Is there an oxygen atom? If there is an oxygen atom, it means that the presence of this oxygen atom, which is very electronegative, it is an electronegative atom, it is going to result in a polar covalent bond, and henceforth this will be uncharged polar amino acid. Then if the answer to all the three questions that you ask are no, then it means that you are looking at a non-polar amino acid. It means that you are looking at a non-polar amino acid. We did look at talk about the polarity of the side chain in that vein, that it affects its solubility. And then we also said functional groups affect their reactivity. that amino acids or other proteins will take place. We also talked about essential amino acids. We say that these cannot be synthesized in the right amounts. So we obtain, or organisms obtain these from the diet.
No.
So they obtain these amino acids, or we obtain these amino acids from the diet. So these are what we call essential amino acids, the essential amino acids. Before we start looking at the peptide bonds, before we start looking at the peptide bonds, which is where we are beginning to look at today. Under the groupings or categories of amino acids, we also did talk about special amino acids or amino acids with special functions. What are some of those amino acids that we mentioned that have got a special function? Guys, amino acids with unique properties. Anyone who remembers? So remember I said there's one of the examples we gave was proline. We gave an example of proline. And we said that proline is the only alpha amino acid. This proline causes kinks in chains. So when proline occurs in a chain of amino acids, it's going to cause kinks or bends. Then we said that cysteine has a special function of linking chains together. And methionine is a special amino acid. Why is methionine a special amino acid? What unique properties does it have? Based from previous other lessons that we did.
Is that protein? Is that the beginning of the protein?
Thank you very much. Yes, so that's true. So methionine is often the first amino acid in a chain of amino acids. Remember the codon for methionine is the start codon. Yeah, so methionine is also a special amino acid. It's a special amino acid. Okay, looking at peptide bonds now. We did look at the process of condensation and hydrolysis extensively in our lesson on macromolecules. So if you haven't yet listened to the recording, you can just listen to the recording on macromolecules to be very helpful for you to understand the macromolecules, the individual macromolecules. We looked at the process of condensation, in which we said that macromolecules or polymers are formed by the process of condensation. And hence in the process of condensation, water is given out as a product and bonds are formed. Bonds are formed. During the process of hydrolysis, this is the breaking down of polymers into their individual monomers. So the bond is broken. So here there's a reaction between a hydrogen atom and a hydroxyl group of two monomers to form a bond. This is the same bond that, or a similar bond that we saw in carbohydrates. What bonds do we find in carbohydrates? Guys, in carbohydrates, what bonds do we find? Which are similar to this, or an example of bonds that are formed through a condensation reaction. Guys, the bonds that we find in carbohydrates, what are they called? They are called glycosidic bonds, isn't it? Right?
Yes, sir.
Yes, they are called glycosidic bonds. So glycosidic bonds in carbohydrates. However, we have peptide bonds in proteins. So remember, in addition to the R group, we found that each amino acid, as we said, when ionized, becomes a dipolar ion. So it has a positive amino group at one end and a negative carboxyl group at the other end. So what will happen is that the amino and carboxyl groups on a pair of amino acids can undergo the process of dehydration or condensation, the one we're just from describing, to form a covalent bond. So in this case, since these are amino acids that are being linked, we call this bond a peptide bond. Unlike in carbohydrates where we called it a glycosidic bond. So it's called a peptide bond. So when you look at a protein is composed of one or more long and branched chains. So it's going to be containing one or more long chains and branched chain. And that each chain is what is called the peripeptide. And that chain is composed of amino acids that are linked by the peptide bonds that we've talked about. So this is the same process that we are describing. This is a process of dehydration. It's a process of dehydration. So remember, it's a reaction that is taking place here. The hydroxyl group of one amino acid and the hydrogen atom of another amino acid. So we have the hydroxyl group from the carboxyl functional group. So what is reacting are the two functional groups to form the peptide bond. So the hydroxyl group from the carboxyl functional group and the hydrogen atom from the other amino acid, from the amino functional group. And water is given out because this is a condensation reaction. What do you notice that has happened here to the carboxyl group? What has happened to the carboxyl group when you compare this and this? Guys, what functional group is this?
Carbonyl.
Thank you. So this is a carbonyl functional group. So we find that when a water molecule is removed during this process of condensation, the carboxyl group here is converted to a carbonyl group here in the resulting polymer. So in this case, since there are two amino acids, it's called a dipeptide. Di means two. So this is a carbonyl functional group. So the carboxyl functional group is converted into a carbonyl functional group. Now, the bond that is formed, has a property, a special property. This peptide bond, due to the electron sharing that happens here, it makes this bond to act like a double bond. Now, if this bond acts like a double bond, what do you think is a property of this double bond? As those that attended the macromolecules lesson, what did we say about double bond? Remember, we started from looking at hydrocarbons, we looked at single bonds, we looked at double bonds and triple bonds. What properties did we say does the double bond have? Has any idea or any idea from anyone? Okay, so remember we're saying this double bond, when these two amino acids are linked by such a bond, they are not free to rotate around this NC bond, the nitrogen carbon bond linkage. Because as I've said, this peptide bond has a partial double bond character. Now this is very different from the NC and from the single bonds. From the single bonds, the CC bonds that are attached to the central carbon of the amino acid. This is not the case. Because of this lack of rotation about this peptide bond, this is one of the factors that determines the structural character of the coils and other regular shapes that are formed by chains of amino acids. So this is going to determine the structural character of the chains of amino acids. Remember, I did say that the double bond, we did say that the double bond resists rotation because a double bond is made of a pi bond, that is called the pi bond, and the sigma bond. And the presence of the pi bond makes this double bond to resist rotation. So that's why this bond, it acting or having a character of a double bond, is important in amino acid linkages or in peptide or proteins in other ways. Is that clear?
Yes, sir.
Thank you.
No, sir, try to repeat a bit.
Sorry?
Try to repeat a bit, just a bit.
So I was saying, when the two amino acids, in this case, we have this amino acid and this amino acid. And we said, through the process of condensation, they are going to form a peptide bond. Now, the formation of this peptide bond that is formed has a special character, due to the electron sharing that happens around this bond. It makes this bond to act like a double bond. Now, double bonds resist rotation. And so the lack of rotation around this peptide bond is one of the factors which determines the structural character of the coils and other regular shapes that amino acids chains form. So that's the importance of this character that is assumed or that is made by this peptide bond. Okay, now, what are some of the key points that we need to note about the peptide backbone or the bonded backbone? So the first thing is the ARA orientation, the ARA group orientation. So remember, these are the ARA groups, the side chains, the ARA group orientation. So one of the key points that we need to note is the ARA group orientation. So we find that these side chains that are present in each residue, they extend out from the backbone. So they are going to extend out, as we can see, they are extending out from the backbone. So this extension outwards, this extending out from the backbone makes it possible for these side chains or the ARA groups to be able to interact with each other. So they're able to interact with each other and also to interact with the water as well. So the ARA orientation, they extend outward, making it possible for them to interact with each other and also to interact with the water. The other key point is the directionality, the direction. So there's an amino there's an amino group at one end and also a carboxyl group on the other end. So the end, this end where we have the amino group, where we have the amino group is what is called the N-terminus. This is the N-terminus. While this end where we have the carboxyl group, where we have the carboxyl group is what is called the C-terminus. So remember we've talked about the ARA orientation or the side chain orientation, them extending out. And then we've talked about the N-terminus and the C-terminus. So which is talking about the direction. So it's talking about the direction. So this end, the N-terminus is also called the amino terminus. This is called the carboxyl terminus or the carbox terminus. So C-terminus and N-terminus. So the other property is that flexibility. Flexibility. So remember I said that amino acids, when they are joined at that peptide bond, there's lack of free rotation. There's lack of free rotation around that peptide bond. However, even though the peptide bond in itself cannot rotate because of its double bond nature, find that this amino acid sequence contains single bonds on either side of the peptide bond, which can rotate. So this means as a whole, the amino acid sequence is flexible. So there's that property of flexibility. There's that property of flexibility. So when these amino acids are linked by peptide bonds into a chain, the amino acids in the chain are now referred to as residues, so that we are able to distinguish them from the free monomers. So that we're able to distinguish them from the free monomers. So now when there are few than 50 amino acids that are linked together in the way that we've described via peptide bonds, that resulting polymer is what is called an oligopeptide. It's called an oligopeptide or simply a peptide. Then those polymers that contain that contain at least 50 amino acids are what are called peripeptide. They're called peripeptide. So find that actually some proteins are able to function just as a single polypeptide, while others are functional only when there are actually multiple polypeptides that are bonded to one another. Now, if we remember when we looked at post-translational modifications, we did say that the shape of a protein is very important as it determines its function. So I am reiterating the same point, that the shape of a protein determines its function. So the polypeptide chains that make up a protein are twisted or folded to form a macromolecule with a specific conformation or a 3D or three-dimensional shape. So it forms a three-dimensional shape. Now, we find that proteins are actually organized in levels. So there are 4 main levels of protein organization. And as we dive into this, we are going to realize that there are various interactions that contribute to how a protein shape is. So we're going to find hydrogen bonds. Sure, we are very familiar with hydrogen bonds. We're also going to find disulfide bridge. Remember when we talked about macromolecules under the topic for functional groups, we talked about the sulfhydryl functional group. And we said that it is found in amino acids or in proteins. So this is the disulfide bridge. Then also have interaction between ions forming ionic bond. And then also the van der Waals interactions, which are simply, they are weak interaction which exist between atoms due to oppositely polarized electron clouds. And then we can also have hydrophobic interactions. So we're going to meet these interactions at different levels of the protein structures, at different levels of the protein structures. So scientists recognize 4 main levels of protein organization. So we have the primary structure, secondary structure, tertiary structure, and quaternary structure. So primary, secondary, tertiary, and quaternary structure. Now, what's the primary structure? Anyone can give it a try? Anyone who can give it a try? So this is simply a sequence of amino acids. So the primary structure consists of simply the sequence of amino acids, which are joined by the peptide bonds, as we have explained. Now, what specifies this sequence? Guys, what specifies this sequence? What specifies this sequence? From our previous lessons, okay, so this sequence is specified by the instructions that are contained in a gene. A gene, the instructions that are contained in a gene. Remember, we defined what a gene is. And we actually said that the gene is going to specify the amino acid sequence. When we are looking at transcription, we said that the gene is that segment of DNA that codes for a complete polypeptide. So a gene will code for a complete polypeptide. So these instructions are specified by genes, So before we continue, we can just log out and log in back.